Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjun classes. Dear student, we are in a continuation of the chapter that is Anatomy of Flowering Plant in Botany. In few previous lecture, we have already talked about tissues, different types of tissues, their functions, their arrangement in the form of tissue system, and the different types of tissue system we have completed. Now, these all things are there, but their arrangement vary in monocot as well as dicot. And in case of monocot and dicot also, it vary in the structures of root, stem and leaf. And that comparison we need to discuss now onwards. So today we are going to discuss anatomy of dicot and monocot root first. Then we will discuss about the dicot and monocot stem. Then anatomy of dicot and monocot leaf. If we talk about first dicot and monocot plant anatomy, for a better understanding of tissue organization, to understand in a better way, it is convenient to study the transverse section of root, stem, and leaves. So when we take a transverse section, we are able to observe all the arrangement of different tissues, and by observing this tissue arrangement, we can differentiate between this internal tissue arrangements of root, stem, and leaves, and that help us to understand anatomy in a better way. So anatomy we are going to discuss in a transverse section of dicot and monocot, but in dicot we will discuss about the root, stem, and leaf transverse section anatomy. Same way, the root, stem, leaf transverse section anatomy in monocot. Now, if we talk about first TS of dicot root, first we are taking the transverse section of dicot root. When we take this transverse section, the internal tissue organization is as follows. The internal tissue organization is as follows. And if you see this internal tissue organization, then you will see that, see, suppose this is a plant, this is the root. We have taken a transverse section of this root, and this in this transverse section we are just observing this part, this many part, only this area we are drawing here. So when we see this structure, you will see the presence of epidermis because it is the root hair. Now we have discussed root hair. Epidermis is also known as piliferous layer or epiblema or rhizodermis. These are the terms which are used for the epidermis of root. Then here you can see the presence of root hair, which are epidermal cell elongation. Then you will see the presence of cortex. The innermost layer of cortex, we call them as the endodermis, which has the Casparian strips, as we have discussed in the ground tissue system. Then you will see the presence of pericycle inner to the endodermis. Then you will see the particular this red color arrangement. You can see that is of the protoxylem and metaxylem. Means it is a xylem, and this protoxylem and metaxylem is nothing but forming a primary xylem. And already we have studied that the first formed primary xylem is known as what? Protoxylem, and later formed primary xylem is known as metaxylem. And you can see here that the protoxylem, which are smaller in size, they are on the outer side, whereas metaxylem, which is bigger in size, that is towards the inner side. So if we talk the protoxylem when it is on the outer side, okay, you can see this particular arrangement. Okay, so here you can see this particular arrangement where protoxylem on the outer side, metaxylem on the inner side, and this arrangement of protoxylem is known as an exarch. We have already studied this. Then you will see the central region, this one, that is a pith. Okay, and the Between the xylem and phloem, between the two xylem, you will see the presence of this phloem radially. So this is the phloem, which you will see the presence in a radial arrangement. So this is the how this particular anatomy is looking. Now let's discuss point one by one. So when we take a transverse section of root, it is represented by, you will see the outermost epidermis, then you will see the cortex, then you will see the stele. Okay, why stele is all the tissue which are presented to the endodermis. And what is endodermis? It is the innermost layer of cortex. So you can see epidermis. Okay, in cortex you will see that hypodermis is absent, which is usually found in case of the stem, but in root it is particularly hypodermis is absent, and its innermost layer is known as endodermis. And as we have studied, all the tissue present inner to endodermis constitute stele. So we have already studied that it include pericycle, vascular bundle, and pith. Okay, so this is the generalization which you can see in this particular root. Now, when we go on talking further, you will see that the epidermis, which is the outermost layer, which is also known as epiblema, piliferous layer, or isodermis, the root epidermis, the many of the epidermal cells you will see that are extending or protruding in the form of unicellular root hair. And you know that the root hair are the epidermal cell elongation and they are unicellular, they are increasing the surface area of absorption. Now, inner to endodermis, you will see the presence of cortex, and cortex consists of several layers, and these several layers are made up of thin-walled parenchyma cells. And as you know, these are thin-walled parenchyma cells which have intercellular spaces, so they are with intercellular spaces. The third is stele, and in cortex also we have discussed that there is an absence of hypodermis, and the innermost layer is known as endodermis. So in stele, the central conducting region of the root consists of pericycle, vascular bundles, and the conjunctive tissue. Okay. Before that, in cortex, we can write that the endodermis innermost layer, its innermost layer is endodermis, and which has a deposition of suberin in form of Casparian strips. Okay, you will see the presence of Casparian strips due to the deposition of the suberin on the radial as well as tangential walls. Okay, now the stele is there. When we talk about the stele, it is the central conducting region of the root, and it consists of pericycle, vascular bundles, and conjunctive tissue, and pith. And pith is also known as medulla. When we talk about endodermis, which we have already talked, which is the innermost layer of cortex called endodermis, it is comprised of a single layer of barrel-shaped cells. Without any intercellular space, they are closely packed. And what are Casparian strips? So we have discussed just that the tangential as well as radial walls of the endodermal cells have a deposition of water impermeable waxy material suberin in the form of Casparian strips. So you will see this is a Casparian strips. They are made of suberin. They are water impermeable. They are waxy material, and they are forming these Casparian strips which are impermeable to water. And you can see here this particularly, this is the cortex cells. Okay, this is the endodermis and this is the pericycle. Now in cortex cells you will see the particularly presence of this. Okay, you will see this is a particularly endodermis. We can say this is the what, Casparian strips. Okay, and this Actually, you can see here, this is the actually this is the Caspian strips here also, and these Caspian strips are not allowing the transport of water. Okay, they are impermeable to water. Now, then you will be wondering that how the water will enter and root into the, because we inside the pericycle there is a presence of xylem. Then how the water will reach to the xylem? Then we have already understood that there are some cells in the endodermis which lake these Caspian strips and that are known as passage cells. So that point we should note down here. Cells of endodermis which lake Caspian strips are known as passage cells. Are known as passage cells. Now, endodermis is not actually a part of stele, but still consists of this pericycle. So next to endodermis, the first layer in stele is encountered. That is the endodermis, and it is made up of thick walled parenchyma, still referred as pericycle. Okay, it is pericycle. Now we have studied in parenchyma also there is a thick walled parenchyma which are known as prosenchyma. So obviously this pericycle consists of prosenchyma. Now, initiation of lateral root vascular cambium during the secondary growth. Secondary growth means girth-wise growth, growth in the transverse section or wood formation is secondary growth. So, during secondary growth, the formation of lateral root means secondary and tertiary root and vascular cambium are formed or arise by the action of this particular pericycle cells or division of pericycle cells. Now, inner to pericycle, you will see that presence of parenchymatous cells which lie between the xylem and phloem. Because xylem and phloem are in a radial arrangement, they are at a different radii or a different radius. And when a tissue which is located between the xylem and phloem, then it is known as conjunctive tissue. So you can see this is the particular anatomy, and in this we can locate it. Okay, so this is the particularly phloem part. Okay, this is the phloem part, and this is the metaxylem, and we can say this is the metaxylem, and here you will see the protoxylem. Now between the phloem and the xylem, whatever the tissue, this blank space is looking, this blank space represent what? Conjunctive tissue. So they are present between the what? Okay, so they are present between this xylem and phloem. Okay, now vascular bundles are represented usually 2 to 4, and 4 2 or 4 xylem and phloem patches. Okay, means they are present in 2 to 4 in number. Usually they can be 2 to 6. Okay, before that, be remember that conjunctive tissue are also, they give rise to part of vascular cambium, although they play a role in formation of vascular cambium, that we can say. Okay, vascular bundle represented by 2 to 4 xylem, usually 2 to 4, and phloem patches, but there can be a 2 to 6 vascular bundle. And when there is a vascular bundle, that is a patches of xylem and phloem, 2 to 6 vascular bundle are seen. Okay, and this 2 to 6 vascular bundle, the technical term which are used, that is diarch to hexarch. Means if diarch, then there means a 2 xylem and 2 phloem. If there is a hexarch, means it is a 6 xylem 6 phloem. So diarch to hexarch mean can be triarch, 3 xylem 3 phloem, 4 xylem 4 phloem, 4 5 xylem 5 phloem. Like this condition is there. If you count this particular numbers and you can see the radial arrangement and you count this vascular bundle, on the basis of that you can determine that which anatomy is of dichotomy. If in school, in experimental part, if teacher asks that this is in showing an anatomy picture and giving you the picture of anatomy just of dicot root, and you want to identify, just you can't see that there is a radial arrangement like phloem and xylem are arranged radially. And one more thing you need to represent, count the number of vascular bundle, then you will be able to answer this question. Now, as we discussed, the little cambium ring developed between the xylem and phloem. Okay, as you can see here, this is the cambium ring, and the pith is small and inconspicuous. Pith is centrally located, and pith is, you can see, the small and inconspicuous, means clearly not visible. Okay, so these things are seen in case of the particular one. Okay, I hope you understood this. And in vascular bundle also we have discussed that radial arrangement is seen in the case of this. So the pith is small or less developed, inconspicuous, or we can say clearly not visible. Why it not visible? Because it is small. We can say less developed. Okay, this many things we can say about the particular. Okay, so this is the particularly anatomical arrangement of the different tissues in the, we can say, dicot root. Now let's talk about another one, that is monocot root. Now when we compare this monocot root from the dicot root, here also you will see the presence of epidermis, root hair, cortex. Obviously, hypodermis is absent. The innermost layer is endodermis. Inner to endodermis, you will see the presence of pericycle tissues, and then you will see the presence of phloem and metaxylem, protoxylem. This metaxylem is inner one. See, this is the large size, the metaxylem, and protoxylem is small size. These are forming a primary xylem, and this primary xylem is also showing the exarch arrangement because protoxylem is on the outer side. So growth is taking place towards the center, and in the center you will see the presence of pith. Okay, now here also conjunctive tissue are going to be present. Okay, but you can see if you count the vascular bundle, you can count 1, 2, 3, 4, 5, 6, 7, 8. The 8 vascular bundle are there. And how much was maximum in case of the dicot root? There was 6 maximum can be possible in case of the dicot root, the number of vascular bundle. But here number of vascular bundle you are seeing that 8 are given. So might be this is the difference which can help us to differentiate this monocot root from the dicot root. So in monocot root, dear student, the anatomy of monocot root is similar to the dicot root mostly, but it has epidermis, cortex, endodermis, pericycle, vascular bundle, and pith as given in the dicot root. So as compared to the dicot root, which has fewer xylem bundles, means usually 2 to 4, but maximum how much can be there? 6 in dicot root. But when we talk about the monocot root, there are usually more than 6 xylem bundles in the monocot root. We can say 6 xylem bundles or 6 vascular bundles, we can say. Means 6 xylem and 6 phloem are there. Okay, so we call it more than 6, more than exarch condition is seen in case of the monocot root. As in diagram also we have seen that there is a 8 vascular bundle are given. So monocot root do not undergo any secondary growth because in monocot root the cambium is absent. So cambium is absent, due to this the particularly they cannot grow in the what we can say secondary growth or thickness wise growth or wood formation. Okay. Now, so this is the thing where you can see this particular point. So by seeing the internal structure of root, which is similar to direct except some differences, the differences which we have just noted, and that are number one, number of xylem bundles, or vascular bundles, if you see, they are more than 6, or we can say more than hexarch. So it is a polyarch condition is seen in the case of the monocot root anatomy. And by this counting the number of vascular bundle, you can differentiate the monocot root from the dicot root. Second, you can see differences from dicot root in monocot root. Pith, as we see here, it is large and well developed, or we can say conspicuous, means visible in case of the monocot root, because secondary growth is absent. And then when we talk about the third thing, that is xylem vessels, they are here appear circular or oval in transverse section. And the one most important point that monocot root do not undergo secondary growth. So this is the thing how we can see the particular differences of monocot root from the dicot root. And that is all about what? Anatomy differences between the dicot and monocot root. Okay, I hope, dear students, you understood this anatomy differences between dicot root and monocot root in a very well manner. Thank you.